In the interest of time, I think we'll move on and on to our final speaker of this session, Fitzwilliam Seibertz, or Will. I think I can almost call you Dr. Will, but not quite yet, right, Will?
Will, yeah, that's right, almost there.
Will is doing his PhD at the University Medical Center Göttingen with Professor Niels Voigt. And Will's been using the synchropatch during his PhD to study stem cell-derived cardiomyocytes and also primary cardiomyocytes at high throughput using automated patch tram. And he's going to present some of his work today in his talk entitled APC in the Atria, an overview of high throughput cardiac chamber-specific electrophysiology. Okay, thanks, Will.
Cool, thanks, Ellie, for that nice introduction. Yeah, exactly right. So I'm the PhD student in a basic science research lab. And the main focus of our group is actually to study the underlying molecular mechanisms of atrial fibrillation, which of course is a highly common cardiac disease, which is unfortunately associated with very clinically poor outcomes for patients. And so I'm going to talk today about how high throughput electrophysiological techniques, such as automated patch clamp, have really benefited our lab in the rapid characterization of atrial specific cellular electrophysiology. So just for a tiny little bit of background, atrial fibrillation is characterized by the loss of function of the sinoatrial node. Now, normally in a healthy patient, this node acts as a pacemaker and ensures steady and smooth propagation of excitation across the atria and then eventually to the ventricles. However, in this disease, the loss of function by the pacemaker is facilitated by the development of very high frequency ectopic beats across the atria, which cause the atria to, of course, contract rapidly and fibrillate. And this constant activity actually stimulates the cardiomyocytes of the atrium to develop different electrophysiological characteristics. And this then, unfortunately, promotes the worsening of the disease. So the cells change, their electrical currents are remodeled, and the disease gets worse and worse. These changes are known as electrical remodeling. And as a very small example here, taken from cardiomyocytes directly from healthy patients and patients with atrial fibrillation, we can really see a decrease in the L-type calcium current density in the atrial fibrillation patients as a result of this remodeling. So when we look at the history of antiarrhythmic drugs and atrial fibrillation treatments, we can actually see that most of the basic science research work into the mechanisms, the cellular or molecular mechanisms of atrial fibrillation has only really taken place in the last 20 years. This is in stark contrast with the vast majority of rhythm control drugs that have been developed to treat AF for patients, which of course have been developed over the last 40, 50 years. And we assume that this difference or discrepancy is based on the fact that these drugs were developed, potentially with a very low translational therapeutic index as well. These drugs have been developed more on observational readouts or recordings rather than a deeper understanding of the molecular mechanisms actually going on. And so we believe that currently this discrepancy between the available drugs and the development of new compounds that have gone on and this increase in the interest in studying the mechanisms is due to an incomplete set of models with which we can actually accurately investigate the mechanisms of atrial fibrillation. What I mean by that is that much of the mechanistic insights that we've achieved over the last 20 years has been done by using animal models. And animal models, of course, contain very highly species-specific differences in their electrophysiology and are therefore maybe not so appropriate for translation into human patients. And so I'd like to talk today about an alternative model with which we can study human-specific atrial electrophysiology. And by that, I'm talking about atrial subtype-specific induced pluripotent stem cell-derived cardiomyocytes. So these iPS cell cardiomyocytes, they've been a well-established technology in the last 20 years. They very nicely allow somatic cells from humans to be reprogrammed to display a pluripotent state. And from then on, they can be further differentiated into any major cell type in the human body. For this timeline that I'm showing on my slide here, this is an overview of the differentiation from iPS cells into cardiac cells, or iPSC-derived cardiomyocytes. It's a 20-day process. We involve many different transcription factors and many different constituents that we give to the cells at various time points to really facilitate their development into real cardiomyocytes. They contain contractile machinery, they contain appropriate electrophysiology. They're a very nice model for us to use in the lab. Now, usually it's quite well known that using standard protocols, iPS cell-derived cardiomyocytes contain a very wide arrangement of subtypes. You've got some ventricular types in there, you've got some atrial, you've perhaps got some nodal subtypes. And so in order to really shorten this spectrum and to isolate only atrial subtype-specific cardiomyocytes, we're employing a relatively new technique, which simply involves the application of retinoic acid early in the differentiation process. Now, this is a vitamin A derivative. It's been recorded to be increased during embryogenesis when the primitive heart fields are first developing. The fields that eventually form the atria seem to have a higher concentration of retinoic acid in the developing embryo. And so if we actually put this onto our iPS cells early in differentiation, we can actually stimulate the development of a very nicely pure population of atrial iPS cell cardiomyocytes at the end of our differentiation process. Now I'm showing you some nice staining here to illustrate what I mean. In the green stain, we're looking at ventricular markers, in this case, ventricular myosin light chain. And you can see in the cells that remain untreated with this extra addition, we've got a high population of green cells, high population of ventricular cells, very nice. However, once we add the retinoic acid and stain for atrial specific markers, shown here in red, we can really see that a huge population is obvious, showing the atrial isoform of myosin light chain as well. So using this technique, we're reasonably happy to assign these two different cell subtypes as either ventricular subtype specific without the treatment or atrial subtype specific with the treatment. And now, of course, my job in the lab has been to begin our investigation of these atrial cells, to probe their electrophysiology, to see if they are adequate as an in vitro model for studying AF. And this is, of course, where our very nice automated patch clamp system, the SynchroPatch, comes in. That requires not so much introduction, but Nanian have developed this lovely high throughput device in which cells can be automatically placed by a robotic liquid handler moving across the screen there into the recording module. And this module contains 384 individual channels by which up to 384 cells can be probed for their voltage characteristics or their current characteristics as well. And so in a simple batch of experiments, I split a plate into ventricular iPS cell cardiomyocytes and atrial iPS cell cardiomyocytes and began to investigate their electrophysiological characteristics. And I'll just provide a little run through of what I've been able to do so far. Measuring action potentials, of course, measuring the voltage characteristics, these cells did show us a very nice subtype specific difference in their action potential morphology. Now, this is the same as in human patients. Ventricular cardiomyocytes do tend to give us much longer action potentials with a more obvious plateau there. And that's exactly what we see in these iPS cell cardiomyocytes as well. We see a longer action potential for ventricular iPS cell-derived cardiomyocytes and a shorter, more triangular shaped action potential for our atrial cells as well. So this was a very nice first step. These cells obviously have some atrial specific markers, and so far they show us very nice atrial looking action potentials as well. Now, the beauty of an automated system such as the SynchroPatch is that it actually allows for the measurement of many electrophysiological parameters during one experiment. And so for us in the basic research area or basic science area, that's very exciting to cut down on time and squeeze out as much data as we can from minimal cellular material. And so once I change amplifier configuration and change a few other bits and bobs, I start to measure the sodium currents as well. This is just nav 1.5 at the moment. And we see very nice, also very clear subtype specific differences between these two cell lines, which is also exactly what we expect to see in human native or primary cardiomyocytes as well. So we're able to get very nice voltage dependent characteristics here and very nice and expected NaV 1.5 currents as well. Moving on, we wanted to really investigate as much as we can to get a full panel of general electrophysiology from these cells. And we also saw very nice subtype specific differences in the L-type calcium current, CAV 1.2 as well. Much larger density in the ventricles and a smaller density in the atrial cells, which is also exactly what we expect to see from or compared to the primary methods of methods of investigation. Now, lastly, or almost lastly, we also wanted to look at the IK1 currents. This is the Kerr 2.1 channel. And this is very important for these cells to have some kind of stable resting membrane potential. So it's been a hotly debated topic whether these iPS cell-derived cardiomyocytes actually have IK1. And then as the technology has developed over these last few years, it's been relatively clear that indeed IK1 can be robustly measured in these cells. And we can even show that they show subtype specific differences as well, where if we compare the inward component of IK1, what I'm showing here is the current readout during a depolarizing voltage protocol. So we're essentially looking at a real-time IV curve of Kerr 2.1 here. And we're able to see subtype specific differences here as well. So we've got larger IK1 in the ventricles and smaller IK1 density in our atrial cohort as well. Now, something a bit more exciting is the identification of only atrial specific currents. So far I've just shown a nice panel. Yes, it seems to be smaller in the atria, just like it should be, very nice. However, the atrial or the cells of the atrium also contain atrial subtype specific ionic channels as well. These channels are absent in the ventricular cardiomyocytes and therefore show a very nice overview of, sorry, just turn those monitors off. Very nice overview of the real atrial specificity of our cellular populations. So once we stimulate these atrial specific currents, what I'm talking about here is the acetylcholine activated inward rectifier current. Now this is only shown in the atria of primary models. And it's very cool to be able to see that in our iPS cells, particularly measured with automated patch clamp, once we stimulate these atrial only channels, we see a response, of course, only in our atrial cohort. And that shows an increase in the inward rectifier density upon application of a drug which activates these atrial specific channels. You can see that in the atrial cells, this presented as an increase in current density. However, no change was recorded in the ventricles as well. And so that was really nice to be able to validate our atrial cohort as really containing atrial specific electrophysiological characteristics. Now we feel on our side that the synchropatch also gives us a very unique opportunity to use this method of screening for atrial specificity as a simple way to give us an overview of how pure our atrial populations actually are. So in a separate experiment here, what I've done is the same experiment, and I've simply quantified the response to carbachol in both ventricular and atrial preparations. If the cell presents as a red box here on the screen, that indicates that it is showing positive for this IK-ACH atrial specific current. The black box indicates that it's negative. And so we can see that in the atrial cohort, the response to carbachol was up to 78% of recorded cells. And that essentially means that within this cohort here, we've measured a atrial purity, as it were, of 78% and a ventricular, let's say, contamination of 22%. And that's also a really nice kind of elegant screening method that we've started to use in our lab, just as a separate testing method to really validate the atrial subtype specificity of these cells. For the other basic scientists in the audience, at least, we also took these over to our colleagues who did a flow cytometry analysis on only atrial cells, and they actually found quite similar percentage purities for the atrial structural isoforms of myosin light chain as well. So an overview of the same batch revealed an atrial, or let's say atrial positive cellular cohort of 75%, which lines up very nicely with our patch clamp data. So we were very excited about this new addition to our lab as a way to really nail down this atrial phenotype and ensure that we have an atrial specific model that we can use for future measurements. So I also want to quickly highlight, for me as the PhD student, I also want to quickly highlight the amazing, intense increase in data throughput that the SynchroPatch adds to our lab. Because all of these data that I've shown today, all of those little points and different currents and everything like that, that took me one day to achieve, which was after some years before we got the device of me sitting on the manual patch clamp rig. That certainly would not take one day. That would be more like 1 year. And so as an intense endorsement of high throughput systems for validating IPS cell electrophysiology, we couldn't be happier with the SynchroPatch in our lab. We've only had it for maybe just less than a year at the moment. What I've shown you today, that just simply came from one batch of ventricular cells, one batch of atrial cells, and then 2 plates, one for the experiments and one for the screening. So that was a very satisfying process for us to be part of. Now, as a little bit of preliminary data, just to bring this full circle, now that we've validated our atrial cell line, therefore we can actually begin the real study of atrial fibrillation mechanisms. And so the next part of this project involves the high frequency pacing of atrial cells in order to see if we can replicate the electrical remodeling that occurs in patients with AF, or with atrial fibrillation. In order to see if we can replicate that in vitro, we simply tachypace these cells. We have one group exposed to normal frequency or sinus rhythm electrical pacing at 1 Hertz. And then we have another group exposed to 3 Hertz tachypacing, or let's call it the atrial fibrillation group. Now, these preliminary data that I'm about to show, that's happened only after 24 hours of stimulation, but we were able to really robustly see evidence of this electrical remodeling, so-called atrial fibrillation associated electrical remodeling in our atrial cohort, where we did see this characteristic reduction in L-type calcium current only in the 3 Hertz paced group, or in the atrial fibrillation group. And that's really exciting. This is still preliminary data at the moment, but this means that we really have a robust model which replicates the very specific cellular conditions that are experienced by atrial fibrillation patients, and therefore gives us a very robust model to be able to investigate these mechanisms further, and of course develop future potential therapeutic targets or treatments as well. So that's where this project's heading, and that's an example of how the SynchroPatch can be implemented in a more basic science or academic setting in order to further some, hopefully soon, future generation of antiarrhythmic drugs as well. That was probably a little bit short there, but I'd like to end it there, and thank you all for your attention. Big thank you to my group, my boss, Professor Niels Vogt, and the other groups around the Göttingen campus who have helped us out with various other aspects of developing these cells and testing these cells. And of course, our very, very deep gratitude to Nanian, who have helped us immensely in getting this fantastic device up and running. So with that, I'd like to stop there, and I welcome any more questions. Thank you.
Thank you so much, Will. That was a wonderful talk. Really nice validation of your atrial cells. We have a couple of questions, and I see that Adam has his hand up. So I will go to Adam first of all, if you'd like to ask your question.
Yeah, thanks, Will, for a lovely study. I know how hard those experiments are, so congratulations. I've got a few real quick questions. You can just answer with sort of one-word answers if you want. But what sort of success rates are you getting for your atrial versus ventricular cardiomyocytes on the synchro patch?
Yeah, so I can answer that pretty quickly. We're getting absolute success rates for the actual currents between 30% and 10% for the overall cohorts. Ventricular cells are more successful than the atrial at the moment. We're seeing if we have to differentiate for ventricular atrial, we're probably looking for sodium, let's say, we're probably seeing around 30% success for ventricular and around 25% for atrial. So we're always a little bit of a step down in the atrial category. But overall, we're around the 30% mark.
And doing anything special in the dissociation, or what enzyme are you using?
We're actually, I haven't told Nanny in this, but we're actually ignoring a lot of their nice recommendations. We're using really harsh trypsin enzyme-based digestion processes. I can see Ali rolling her eyes there. I haven't had too much success with gentle digestion procedures. I'm not sure exactly why. And so in the chase for just speeding things up, I've gone back to our usual trypsin-based digestion, and it does seem to be working reasonably well to give us those success rates. I'm sure with further optimization, we can get that much higher as well.
Yeah, I think our experience probably correlates with that. We ended up back at trypsin. One more real quick one then. With your tachy pacing, do you see any overt arrhythmias in any cells, or just the remodeling?
At the moment, we actually just see the remodeling. So we don't see any clear arrhythmias such as, or triggers such as after early depolarizations or delayed after depolarizations. We simply see the maintenance of a higher spontaneous rate after the pacing.
Yep.
And the remodeling has been our major finding so far, as a sense for some kind of arrhythmic phenotype. Okay,
great. Thank you. Great talk.
Thank you.
Okay, thanks, Adam. Thanks, Will. We have a couple more questions. I have one from Marcus, my colleague. He says, fantastic data to see. Thanks so much. Are you planning to differentiate iPSCs to pacemaker cells?
That's a good question. And from my side, from our side, no. So we currently, there are protocols out there, but we have very little experience in generating specialized protocols to only produce nodal cells at the end. So from our side, unfortunately, we won't be doing that anytime soon.
And from Jamie Vandenberg, we have great work. Will, in addition to APD90, did you measure the resting membrane potential? And if so, was the average value less negative in atrial compared to ventricular myocytes, consistent with the differences in IK1?
That's a very good question. At the moment, with the synchro patch, we are using quite high holding currents to get these cells at a hyperpolarized enough level to initiate an action potential. And so based on the data that I've created here, we saw no difference in resting membrane potential between the two subtypes. However, I didn't measure absolute resting levels in the absence of holding currents. So when using other models, when we put these cells into three-dimensional tissues, we do see changes in resting membrane potential consistent with the IK1 values. So outside of the automated system, we do see a generally more depolarized resting membrane potential in atrial cells. However, not in the data I showed today.
Another one we have from Bogdan. Did the atrial or ventricular stem cell derived cardiomyocytes feature spontaneous pacing in culture?
They did, yes. So in the absence of any other intervention, so we weren't culturing them under any special conditions, they showed spontaneous beating that was consistent with other literature. So we saw quite high frequency spontaneous beating in atrial compared to ventricular. However, both were below 1 Hertz in terms of their basal spontaneous rate. So atrial, we saw around 0.9, 0.8 Hertz spontaneous beating rate, and ventricular were a lot lower, around 0.5 Hertz spontaneous beating rate.
Okay, and another one from Shinsuke-san. What's the mechanism of LTCC current decrease?
That's a very good question. So it's actually a calcium-dependent mechanism. And what we're seeing is that once you're stimulating these cells so much, they're experiencing a huge amount of calcium influx during their rapid contraction relaxation system. And this actually activates an NFAT-dependent signaling pathway, which downregulates L-type calcium channels in the membrane. We also can see, or others before us have also seen, that it's also a phosphorylation-dependent process. So as well as L-type calcium channel downregulation or trafficking away from the membrane, we're also seeing a decrease in phosphorylation of these channels. And decreased phosphorylation means we see less current coming through. I've just the other week, I also tried putting a phosphatase inhibitor on these cells. So we wanted to increase the phosphorylation. And we saw an almost reversal of this electrical remodeling. So we saw the L-type calcium current get bigger and back down to normal levels. So there's a bunch of different mechanisms in play, some involving downregulation of the actual channel and some involving phosphorylation-dependent mechanisms as well.
Hey, great. Thank you so much. Are there any more questions? I don't see any. So, okay, well, thank you, Will, and thanks to all of our speakers from this morning's session. This was a great mixture of different aspects of cardiac research.